I'm Randy Niedz, and I work at the U.S. Horticultural Research Laboratory in Fort Pierce, Florida. SIVB has scheduled this workshop because the in vitro community has expressed a lot of interest in the design of experiment or DoE approach to experimentation, an approach that was almost single-handedly developed by Ronald Fisher in the 1920s. This workshop will include 3 sections. This first section, we want to provide some context of what DoE is, how it differs from the conventional approach to experimentation, and some of the key principles that make up the DoE framework. And then I'll talk a bit on software requirements. In the second section, Ms. Wynn will provide a walkthrough of an in vitro project using DoE. Her walkthrough will show what DoE experimentation looks like, from defining the problem, planning the designs, collecting the data, analyzing the data, and interpreting the data. And then the third section will be a Q&A session. Here are some DoE textbooks in my library. A page count shows that there's been quite a bit written about how to conduct an empirical investigation. And these books describe many experiment types, so not every page applies to every test. But there is a great deal of material in any of these books that would apply to any given experiment. These and many similar books are readily available, and adding a few of them to your library is worth doing and would pay dividends. Probably the most basic question is, why DoE versus, say, conventional experimentation? The short answer is that it's much more efficient than testing one factor at a time. For a longer answer, let's look at this a bit more closely and ask, what is the product of a research laboratory? One common answer is that the product of research is something that we call data. And I think of this as the industrial model of research. The quality of the product is enhanced by minimizing and stabilizing the product's variability. The productivity of the laboratory is synonymous with a high volume of the product. The more data, the better the productivity. The factory model is not an appropriate framework for a research laboratory. And the reason is that laboratories do not produce an industrial commodity. Laboratories produce a scientific research product. And that product is not data. It is knowledge. And we bear the expense of research for only one reason, and that is to learn something that we do not already know. Now, this means 2 things. If we get the wrong answer, the volume and quality of the data is irrelevant. Also, if we get the right answer, the volume and quality of the data are irrelevant. So all notions of quality and productivity change when we recognize that knowledge is our product and not data. Let's talk about the impact on quality and productivity. Why maximize data volume if data is not the product? Why focus on data quality if data is not the product? DoE integrates true quality and productivity measures within the experimental process. And this means that quality and productivity are no longer external considerations. Quality and productivity are assured through the design of the experiment itself. What happens to quality and productivity when data is not the product? A productive DoE experiment minimizes the volume of data required to adequately predict system responses. As mentioned previously, the product of a DoE experiment is knowledge. But what is knowledge? We know a system when we can adequately predict responses such as rates of shoot multiplication, rooting, embryo formation, transformation, gene editing efficiencies, plants established in the soil, etc., for any combination of independent variables that interest us, such as media components, environmental conditions, and genotypes. When we know the system well enough to adequately predict its behavior, we can stop expending resources to test. And this often happens early in DOE experimentation. So here's another difference between DOE and conventional experimentation. Conventional experimentation ends when the allocated resources are exhausted. A DOE experiment is over when we know the system well enough to adequately predict its future behavior. Savings of a factor of 2 or more in cost and time, and similar reductions in uncertainty are not uncommon with this change in focus from data collection to knowledge enhancement, and a factor of 10 is not unusual. Okay, let's talk a bit about how DOE differs from conventional experimentation. Conventional experimental methods typically employ the one factor at a time approach, or what's called the OFAT approach. This is an exhaustive enumeration strategy in which a large number of independent variable combinations are systematically configured. The emphasis is on high data acquisition rates to cover the large data volume needed to physically set every independent variable combination of interest. It's relatively expensive because more data are required than is usually needed. The emphasis on speed forecloses certain options to enhance the quality of the experimental result. OFAT designs require no training. This is what is done at grade school science fairs. We come out of the womb knowing how to do OFAT testing. OFAT testing is the most costly method of experimenting, and it's generally too expensive to sample the design space with OFATs. Fisher talked quite a bit about the merits of testing one factor at a time. He says that this simple formula is not very helpful for research. And an example where he is quite blunt. If the investigator in these circumstances confines his attention to any single factor, we may infer either that he is the unfortunate victim of a doctrinaire theory as to how experimentation should proceed, or that the time, material, or equipment at his disposal is too limited to allow him to give attention to more than one narrow aspect of his problem. And Fisher talks quite a bit about this, and this is just a sample. The DOE literature generally describes why OFAT experimentation is inefficient and costly. As a general approach, this is true. However, I want to mention where OFAT testing is sometimes a good choice. Where OFATs can help is when there are inference subspace boundaries, or ISBs. Now, that sounds like a complicated term, but these are simply sharp changes in a response, and this is an ISB. So OFATs can be very useful to get a preliminary, a lay of the land before we start DOE experimentation. And I want to give you an example because I think there's some things in plant tissue culture where ISBs are important. And these are the trace metal experiments. And I'm going to give you a couple examples here of how we used OFAT testing to get a look in terms of range finding of where we should be experimenting. So here's a couple examples, one from Gerber daisy and the other from Brugmansia. Before starting mineral nutrient optimization experiments in Gerber daisy, we wanted to know the range that we should be working in regards to the microelements. We simply didn't know. And so we did an initial scan where we ran a log series from 0 to 1 millimole for a number of the microelements. So here are the results for iron and zinc. The MS level of iron is 0.1 millimole, and for zinc it's 0.037 millimole. And as you can see, the regions to consider in follow-up experiments are quite clear. And it's in these regions where we would then use response surface methods to identify optimum levels. Here are the results from Gerbera and Brugmansia to manganese. In both species, the range to sample is quite a bit lower than MS. Manganese is at 0.1 millimole in MS, but the scan indicates about 2 logs lower may be the region to examine. I've talked to some very good DOE statisticians and practitioners about using OFATs for range finding before starting a DOE project. They not only agreed that this is where OFATs can be very useful, but they admitted that this is how they use OFATs. Let's talk a bit about how the one factor at a time, or OFAT method, actually works, and to see why it is so inefficient. Step 1 is to identify the independent variables. For tissue culture, this would include media components and any external factors. Step 2 is to develop a wish list of all the levels that you want to set. What I list here is what this list can typically look like. It might actually be much longer. Remember, this is a wish list. Step 3 is to face the reality of resource constraints. Our wish list requires over 4 million treatment combinations that would have to be run one at a time. Step 4 is then we consider ways to reduce the resource requirements. Perhaps we would accept fewer auxins and cytokinins, or perhaps coarser increments between the settings. So this reduced list has a little over 8,000 individual combinations. This is a big improvement, but it's still way beyond our resources. So we move to step 5 and we reduce the scope even further of the study so that we can conserve resources. And so we limit the number of cytokinins, we fix temperature so it's no longer varied, etc. This reduced scope experiment has 32 individual combinations. Congratulations, we are now within budget. The 13 factors that were in our wish list is actually a 13-dimensional design space, or as I mentioned previously, you'll see this referred to as inference space. Here's a diagram showing what we wanted compared to what we could afford using OFAT experimentation. This, of course, is not to scale, since the red area would not even be visible, since we started with over 4 million combinations and ended up with 32. In fact, to see that red area, you'd probably need an electron microscope. There's simply way too many factor combinations to set them all one at a time. But it's worse than that. And in fact, it's potentially a lot worse. If there are interactions, then we may produce relatively meager results, since OFATs cannot detect interactions. And I do not have to tell you that interactions in plant tissue culture are plentiful. There's simply never enough time or money to examine every factor combination one at a time. Wider intervals between the settings is unattractive, and eliminating factors is bad when there are interactions. So what is the solution? The solution is to make every data point work harder. Don't just change one factor at a time. Change many factors at a time. And look at how changes in many different factors affect the response with every data point. This means that every data point will contain information about every factor. And then we separate out the individual effects in the analysis phase of the experiment. And I'm going to use a simple experiment to illustrate how this works. So we will illustrate the tactics that DOE uses using the monkeys and gold double pan balance weighing experiment. You are asked to weigh two objects. You have access to a double pan balance and a set of counterweights. The two objects are a brass figurine of the three wise monkeys. When I was very young, maybe 4 or 5 years old, I remember one of these on a dresser at my aunt and uncle's house, and I was fascinated by it. And the second object is a vial of gold flakes from an Arizona gold mine. This is something I guess typically you can get in a gift shop in the gold mines. Now, the amount of real gold is infinitesimal since the flakes are gold plated and the gold is only a few atoms thick. The requirements and cost of the experiment. For the requirements, the results should be reported with a standard error no greater than s over the square root of 2. And this tells us that each object must be weighed twice. Each measurement will cost $1,000, and each measurement takes 1 hour. So how would you conduct this test? The conventional approach of getting data is to measure each object twice. Therefore, the result is 4 measures, or runs, as they're called in DOE. So the balance is used 4 times at a cost of $4,000 and a time requirement of 4 hours. Can we improve on this? Yes, and absolutely we can improve on this. The secret of DOE is that data points are made to work harder than in a conventional test. And the way this is done is that rather than examining one factor at a time, we examine all factors with each measurement. Every data point contains information about all the factors in the experiment, not just one. Here's the DOE approach to weighing the monkeys and the vial of gold flakes. The results are quite a bit different from the conventional approach of weighing the objects one at a time. Only half the time and money are required. 2 measures were used, or runs, so the balance was only used 2 times rather than 4. Therefore, the cost was reduced and was $2,000 rather than $4,000, and the time was 2 hours rather than 4 hours. So with this simple experiment, we got a 50% savings. But what's interesting is that the more factors that are involved in an experiment, the greater this leverage becomes using DOE. Now remember, I said that the effects are separated out at the analysis phase. Here is shown how the weights of the 2 objects are determined. I'm not going to go through this in detail since this is being recorded and is available online, and you can review the details at your leisure. Regarding the precision of the measure, here are the details. Again, I'm not going to go through this in detail, and you can review this online since it is being recorded, but the precision requirement was satisfied. So let's summarize what we learned from the double pan balance weighing experiment, where we compared the conventional approach of single factor experimentation with its emphasis on data acquisition to the design of experiment approach with its emphasis on acquiring knowledge. The DOE design only consumed half the resources, but still met the same test objectives. Every measurement worked twice as hard. Monkeys were weighed with every data point, and the gold was weighed with every data point. Resources were not wasted by weighing each object one at a time. DOE methods improve productivity in more complex experiments in exactly the same way that I just showed you with the double pan weighing experiment. Multiple variables are changed simultaneously to see how they all affect the responses. And then the effects are separated out in the analysis. Now, let's talk a bit more about the analysis phase. There are 3 DOE principles that are used to determine which effects are important, and these are particularly useful when analyzing fractional factorial designs. The term fractional factorials refers to designs that have multiple factors where each factor is set at 2 levels. These are the sparsity, hierarchy, and heredity effect principles. In Wynn's walkthrough, she's going to go through a fractional factorial. And if you're not familiar with fractional factorials, they are probably the most underutilized designs, but they are extremely useful and efficient. As an aside, I wrote a chapter last year on fractional factorials. I included a complete walkthrough of a fractional factorial, and it includes all the calculations. I wrote it this way since the calculations can all be entered and done in a spreadsheet. So this allows people to try and even use fractional factorials without having to purchase software, which can be expensive. While I was preparing for this workshop, I ran across a paper in Nature Methods that really surprised me. It's a paper on 2-level fractional factorials. So it was published in a journal that will only publish what they think is highly innovative and groundbreaking work. The information they present was explained by Fisher in the 1920s and is a core component of any DOE book. It's widely available. I'm guessing the reason this was published, even though the information is widely available, is that they are rarely used and seen in the research literature. And Nature Methods felt it was important to remind the research community of these underutilized but very useful designs. Back to the 3 principles. I took the 15 effects that can be estimated from a full 4-factor 2-level factorial and illustrated them as layers. Main effects on top, then 2-factor interactions, then 3-factor interactions, etc. I sized the boxes to show the relative size of the effects. The sparsity of effects principle states that only a few effects will be active, and the analysis identifies 5 active effects from the 15 total. The effect hierarchy principle states that lower order effects are more likely to be active than higher order effects. The hierarchy principle is an a priori principle. In other words, before we know anything, we think that a main effect is more likely than a 2-factor interaction, and a 2-factor interaction is more likely than a 3-factor interaction, and so on. The heredity principle states that an interaction is more likely if its parent factors are also significant. For example, if BC is under consideration, then it is more likely to be significant if one or both of its parent factors, B and C, or B or C, are also significant. Where this is important is when using the fractional factorials, where effect aliasing occurs. Aliasing does not occur with full factorials, but is one of the features of fractional factorials and must be considered in their use. Understanding factor aliasing is the key to using and understanding fractional factorials. I think aliasing is the thing that makes fractional factorials at first seem quite confusing. And I suspect it's probably why there are not very many studies that use fractional factorials. But that soon goes away once you start using them. In fact, I would say that the single most useful designs when learning DOE are the fractional factorials. Here's what aliasing, and the term is sometimes called confounding, looks like. For example, the two-level fractional factorial designs that are called resolution 4 designs are used for screening factors to identify those with strong effects. The two-factor interactions in a resolution 4 design are confounded with other two-factor interactions. If this were a resolution 4 design, then the significant BC two-factor interaction could also be AD or CD. We simply do not know. But the hereditary principle provides some insight on which interactions are most likely. Wynn will provide a walkthrough of a resolution 4 fractional factorial, and you'll get to see this principle again. Here's a good reference that discusses these principles. There is one other related topic I want to show you, and that is the hierarchical principle that's used when building a model. For example, say there's a significant two-factor interaction of A and B, where the main effect of A is not significant. The hierarchical principle says that the A main effect should be included in the model because it is a parent of the A-B interaction. Okay, I want to say a few things about software requirements because DOE does require software. The designs, data, and model adequacy tests, the analyses, graphing are all done with DOE software. In fact, DOE really started appearing in the research literature once the software became available, and this was about the mid-1990s. There are quite a few commercial DOE software packages. JMP, Design Expert, Minitab are widely used. There are many others. If you look at the commercial packages, they have extensive support, documentation, user communities, etc., etc. These are all the kinds of things that you want when you're selecting a package. I did run across one called Develve. It was $80, and the $80 is to purchase. It's not a subscription fee. So very reasonably priced. I don't know anything about it, but I think it's worth trying. As far as free software, the preeminent package is R. R is extremely powerful. In fact, so much so that it is used by professional statisticians and many in the research community. It's widely supported. There's a lot of documentation. There is a bit of a learning curve. Of course, there's going to be a learning curve on any of these. But if cost is a big issue and you really need to do some of the more sophisticated DOE, R is worth looking at. Okay, with that concludes this first section of the workshop, and I will turn it over to Miss Wynn, who will then provide a detailed walkthrough of two of the most common and widely used DOE designs, a fractional factorial for screening and a response surface design for optimization.